right. So let's move on from Mendel and let's look at some more kind of, let's take a special look at some genetics that are a little bit more advanced than Mendel, we'll say. All right. So Mendel gave us a great foundation, but obviously, as we learn more about genetics and as technology increased, we were able to get a better view of how genetics really does work. So let's see if we can pin here. And move on. All right. So some things we're going to talk about in this video. We're going to look at incomplete dominance. We're going to look at codominance. We're going to talk about sex-linked diseases. And we're going to look at some different kinds of Punnett squares, okay? So moving beyond Mendel, basically we have to talk about the fact that some traits, right, some alleles are controlled in ways that are more complex than the way Mendel described. So maybe there's more than one gene involved, or there is some outside influence on how the trait is passed on. So the first one we're going to look at is incomplete dominance. So the law of dominance tells us that the dominant allele should overpower the recessive allele, and that's the only trait we should see in our phenotype. However, that's not always the case. Incomplete dominance occurs when the dominant allele isn't powerful enough to completely overpower that recessive allele. And so the heterozygous offspring is going to look like a blend of the two alleles instead of just the dominant phenotype. 4 o'clock flowers are a great example of incomplete dominance. Now, normally we would say red is the dominant gene, and so we should use little r for white, but that's not the case here. In incomplete dominance, we're going to use both letters, R for red, W for white, but the recessive one is still going to be lowercase. So big R plants would have red flowers, little w plants would have white flowers, but RW, right, the hybrid, is going to have pink flowers. Now, this genotype is really important because since it's two different letters, it tells us, hey, we're talking about incomplete dominance. Now, these are 4 o'clock flowers. You can see that down here, right, these guys, we have little, let's see, little w, all right? And these guys right here, we have big R. And these pink fellows, though, they're going to be big R, little w. So that is an example of incomplete dominance. Now, another special case is called codominance. And in codominance, more than one allele is a dominant gene. And so here, the phenotypes won't blend, but they're going to both show up as specific colors. We're going to use colors in this example. So we're not going to use colors. Sorry, I'm going to use blood types. So A and B are both dominant. So you've probably heard someone has type A blood or type B blood, or they can have type AB blood. Type O blood is actually the recessive version. So when we look at their alleles, we use I for, I believe it's immunoglobulin, all right, factor A or factor B, whatever. Immuno, there we go. There's a U in there. Anyway, so A, B, and O. Now notice, these are both uppercase. And in this case, we have a lowercase gene or allele. So type A blood can be big A, or big A, little o, because remember, O is recessive. Type B would be big B, or big B, little o. Type AB, you would have one of each, A and B factor. And type O, you would have to have just recessive, right? So this is our a great example of codominance, all right? Now, keep on kind of moving along with these special cases. Sometimes we have traits that are controlled by multiple genes. This is called polygenics, all right? Poly means many, right? And genics is obviously talking about genes. So if you have a lot of different phenotypes, and we're not going to look at any specific ones, but let's say like height in human beings. Human beings can be a wide range of different heights, right? You can be, an NBA basketball player like 6'8". You can be 5'2". height in human beings is a polygenic trait. It's controlled by a lot of different genes, not just one or two. Gene linkage is actually an example of the law of independent assortment being incorrect. So because you can have polygenics, right, where many different genes are influencing a singular trait or a singular phenotype, sometimes those genes are on the same chromosome, which means they're going to get passed down together. They're linked together. Mendel didn't know anything about chromosomes, so we'll cut him some slack and say, yeah, the law of independent assortment is true a lot of the times for simple traits, right, that only have one or two alleles. But in the event that you have a polygenic trait, gene linkage can lead to some traits being passed down together. There are also things called sex-linked traits, and these are traits that are linked to your biological sex, okay? So some of the phenotypes that we see in these, and it's often diseases, so we see in these diseases, are expressed more often in males than females or females than males. And this is going to be present in like different differences in the genders, and sometimes it's going to be normal traits that both genders share. So it can be either or. A great example of this is color blindness. But in order to talk about color blindness, we have to make sure we know the difference between the autosomes, which are the regular normal chromosomes in human beings. You have 22 pairs, so 44, and the sex chromosomes, which is going to determine your gender, your male or your female. There's one pair of sex chromosomes in human beings. They are usually visualized through what we call a karyotype. So when we visualize the karyotype, what we actually do is the scientist goes in and takes a picture of each chromosome and puts them together to make this kind of like slide. And you can see that chromosome number 1 is way bigger than chromosome 22. They kind of get smaller as you go. And the very last pair is the sex chromosomes. So these are our sex chromosomes. And 1 through 22 are our autosomes. All right. Now, this is a normal karyotype. This would be for a male because you have the X and the Y chromosome. The only difference that visually anyway is the Y is a smaller chromosome. If this was a female, you would expect something like that, right? They would both look roughly the same. All right, we would have XX, geez, XX. We'll look more at some karyotypes in a different PowerPoint. Let's go back to sex-linked traits. Now that we know these are traits that are only passed on the XY chromosomes that last that 23rd pair. Sex-linked traits actually only come from the X chromosome. And we usually see X-linked diseases showing up in males way more often than females. And the reason behind this is that males only get one copy of the X chromosome, where females get 2 copies. Why is that a problem? Well, if like I'm a guy, I have 1 copy of the X chromosome. If something is defective on my X chromosome that is a recessive gene, I don't have another X chromosome that might have the dominant version that would overshadow it. So with guys, since we only have 1 X chromosome, everything on the X chromosome gets red. Where would we see this, right? Color blindness is much more common in men than in women. So the color blindness gene is actually passed from parents to offspring by the X chromosome. So normal vision, is dominant. So that's big C. Little c will be the colorblind allele. So a normal female would be big C, big C. A carrier female is big C, little c. So a carrier means this female is not colorblind because she has the dominant regular normal vision allele, but she is a carrier, meaning she could pass on the colorblind allele to her offspring. If you have a colorblind female, you would have to get 2 copies of the recessive colorblind allele on your X chromosome, one from mom, one from dad. So dad would have to be colorblind and mom would have to be a carrier. For men, you only have either normal vision or colorblind. There is no carrier colorblindness in men. So what's this look like? All right, well, let's do some Punnett squares. So here I have a carrier female with a colorblind male. So X big C, X little c. So here is a carrier, daughter, X little c, X little c. This would be a color blind daughter. And I could have X big C, Y, which would be a normal son. Or I could have X little c, Y, which would be a color blind son. So in this case, all right, as far as color blindness is concerned, we have a 50% normal pass on of the allele on the left. We have a 50% colorblind allele pass on, right? So two colorblind kids. So a carrier female and a colorblind son. Now, on the other hand, right, if you have a carrier female and you have a normal male, well, what are the chances of, any of your kids being colorblind? So over here, it was 50%. On this side, we have big C, big C. So this is a normal female. Big C, little c. This would be a carrier female. Big C, Y. This would be a normal son, right? Normal male. And we would have little c, Y. This would be a color blind male. All right. So normal, regular color seeing guy with a carrier female. So she thinks she's normal, right? She, as far as she knows, unless she's been tested, she thinks she's normal as far as like what she can see. She can see. Well, here you have a 50% chance of a boy that's colorblind, right? If you have a boy, so we'll say if boy, it's a 50-50 chance it's colorblind, all right? Because the X chromosome that goes to the son comes from the mom. So you either get that one or you get that one, right? One or two. If you get chromosome 2, you're colorblind. So we see sex-linked traits like colorblind showing up more commonly in men than in women. In the girls here, right, you have a 0% chance of a female that is colorblind, right? You might get a carrier, but she can still see. So in this instance, right, we've got, a much more or a much higher chance that the boy is affected by this than the girl. So that's a sex-linked trait, right? We're seeing it in the boys, not so much in the girls. Now, a lot of different, study has gone into genetics. We've determined a whole lot of different impacts that the pass on of genes can have. And these are just a couple cases, right? We want to know these, but there are obviously more. Last year, or I guess 2 years ago, had a student bring one to my attention, which was pretty cool. I decided to include it. Gonadotropomorphism, all right, in which organisms are half male, half female, which comes from kind of an abnormal fertilization of the egg, is an example of a special genetics case, right? So you can see that half the body of this cardinal is the female and half is the male, right? Same thing right here. Same thing right here in this butterfly. So there are a lot of really interesting cases where genetics can, have an issue as far as how genes are being passed on. You're welcome to research those, but we're really just going to focus on incomplete dominance, codominance, all right? We're going to look at gene linkage, make sure you know sex-linked traits, and in another video, we'll talk about a few other ones, all right? So if you have any questions, let me know.